Before the savannas turned golden, before lions claimed their throne, before humanity took its first uncertain steps, Africa belonged to bears. Not the bears you imagine from forest fables or mountain documentaries, but something fundamentally different. These were creatures that rewrote the rules of what bears could be, where they could live, and how they could dominate an entire continent. They hunted in deserts that would kill modern grizzlies in days. They evolved bodies that defied everything paleontologists thought they understood about bear biology. And then, without warning, they vanished so completely that for over a century, scientists refused to believe they had ever existed at all. This is the story of Africa's forgotten ursine empire, a lineage so bizarre, so perfectly adapted to environments bears should never survive in, that their fossils were mistaken for hoaxes, their skulls attributed to mythical beasts, and their very existence erased from scientific memory. What happened to Africa's bears reveals one of evolution's most radical experiments, and why even the most successful designs can disappear when continents shift beneath them. The phantom ursists of the Rift Valley. The first fossil emerged from a cliff face in northern Ethiopia like a whispered secret from deep time. A partial skull embedded in volcanic ash layers dating back millions of years, discovered by a local goat herder who thought he'd found the remains of a devil. When paleontologists finally examined the specimen in detail, their confusion was immediate and profound. The skull possessed the unmistakable architecture of a bear, massive zygomatic arches for anchoring powerful jaw muscles, a characteristic sagittal crest running along the crown, robust canines designed for tearing rather than slicing. But everything else was wrong. The proportions were distorted in ways no known bear exhibited. The cranial vault was compressed, almost flattened, suggesting sensory adaptations completely alien to Ursine evolution. Most disturbing were the tooth wear patterns, microscopic scratches and pits that told a story of a diet no modern bear would recognize. The scientific consensus held that ursids evolved in Asia, spread into Europe and North America, but never successfully colonized the African continent. Africa had its own apex predators, big cats, hyenas, crocodiles, and there was simply no ecological space for bears to squeeze into. That consensus shattered when a graduate student named Catherine Mbogo conducted a comprehensive morphometric analysis using three-dimensional scanning technology. She compared the Ethiopian skull with hundreds of carnivore specimens from across the mammalian family tree, running statistical analyses that measured every curve, every angle, every subtle feature. The results were unambiguous. This was a bear. Not just any bear, but a highly derived ursid that had diverged from all known lineages, evolving in isolation on the African continent for millions of years. The evolutionary distances were staggering. This lineage had split from other bears before the common ancestor of brown bears and polar bears had even appeared. The revelation triggered a paleontological gold rush. Suddenly, researchers began re-examining fossil collections across East Africa, pulling specimens from storage that had been misidentified or ignored. A jawbone from Tanzania, previously labelled as belonging to an unknown carnivore, revealed perfectly preserved molars with wear patterns matching the Ethiopian skull. A partial skeleton from Kenya, catalogued decades earlier as possible bear-like carnivore, showed limb proportions that confirmed the bearer moved differently from any living ursid. Toe bones from Chad, discovered during excavations focused on early human ancestors, displayed weight-bearing adaptations seen nowhere else in the bear family tree. Within 5 years, paleontologists had assembled evidence for not one, but multiple species of African bears, spanning millions of years and thousands of miles. These discoveries fundamentally challenged our understanding of biogeography and carnivore evolution. The presence of bears in Africa meant that ursids had either crossed from Eurasia multiple times through corridor habitats we hadn't yet identified, or that an ancient bear population had become isolated in Africa early in ursid evolution and developed along completely separate trajectories. But recognizing that African bears existed was just the beginning of the puzzle. The real questions emerged when researchers started reconstructing how these animals actually lived. Their skeletal anatomy suggested lifestyles so different from modern bears that paleontologists struggled to find appropriate ecological analogues. Their teeth hinted at diets that shifted dramatically depending on environment and epoch. Their limb bones revealed locomotion patterns optimized for landscapes modern bears avoid entirely. And their distribution across the continent, from Mediterranean forests to sub-Saharan grasslands to the fringes of the Sahara itself, indicated an ecological flexibility that contradicted everything we thought we knew about ursid habitat requirements. The phantom bears of Africa weren't just evolutionary footnotes. They were major players in extinct ecosystems, and understanding their biology would require paleontologists to completely reimagine what it meant to be a bear. Desert ursids and the Saharan paradox. Bears are creatures of forests and salmon streams, of berry patches and winter dens dug into mountainsides. They require abundant water, dense vegetation for cover, and seasonal food sources that support their massive caloric needs. These requirements are so fundamental to ursid biology that bear distribution maps look like inverted desert maps. Wherever you find true deserts, you find an absence of bears. Except in ancient Africa, where fossils tell an impossible story. Bears thriving in the heart of what would become the Sahara, perfectly adapted to one of the harshest environments on Earth. The evidence comes from a series of remarkable fossil sites in what is now Chad, Niger and Libya, regions that today receive less rainfall annually than Death Valley. But these sites preserve sediments from a time before the Sahara's full desertification, when the region experienced cycles of increased moisture punctuated by increasingly severe dry periods. Within these sediments, paleontologists have found bear fossils associated with environmental indicators that paint a vivid picture of the challenges these animals faced. Pollen analysis reveals landscapes dominated by drought-resistant acacias and grasses, rather than the lush forest bears typically inhabit. The bears themselves show morphological features that make perfect sense only in the context of desert survival. Their limb bones are elongated compared to forest-dwelling bears, with reduced muscle attachment areas suggesting leaner builds. This body plan minimized surface area relative to volume, reducing heat absorption during the scorching daytime hours, while maintaining enough bulk to retain heat during frigid desert nights when temperatures could plunge 40 degrees. Their foot bones show adaptations for traveling long distances over hard, rocky ground, rather than the soft forest floors modern bears navigate. Some species developed relatively larger ears, or at least larger ear openings in the skull, indicating enhanced thermoregulation through increased surface area for heat dissipation, similar to the oversized ears of African elephants and foxes. Dental adaptations reveal even more dramatic shifts from typical ursid biology. While most bears are omnivores with crushing molars adapted for processing both plant and animal matter, some Saharan bears evolved increasingly specialized dentition. One lineage developed teeth with extremely high crowns and thick enamel, adaptations seen in animals that consume large quantities of grit-laden food. Microscopic analysis of tooth enamel shows where patterns consistent with grinding tough desert vegetation, seeds with hard coatings, fibrous roots and tubers, perhaps even the tough leaves of desert shrubs. This represents a dietary shift toward foods modern bears largely ignore, resources available year-round in arid environments even when more nutritious options disappear during dry seasons. But the most telling adaptations appear in their metabolic physiology, inferred from bone microstructure and growth patterns. Desert bears seem to have evolved a different strategy entirely. Their bone tissue shows continuous growth patterns without the seasonal growth lines typical of temperate bears, suggesting they remained active year-round rather than entering extended dormancy. This makes sense in desert environments where winter is actually the most productive season, when cooler temperatures and occasional rains bring temporary abundance. Desert bears had to remain alert and mobile to exploit these brief windows of plenty. Water acquisition poses perhaps the greatest challenge for any large desert mammal. Modern bears require daily access to water, and their kidneys are relatively inefficient at concentrating urine compared to true desert specialists. Yet Saharan bears lived in landscapes where water sources might be separated by 50 miles or more during dry seasons. Analysis of bone chemistry provides clues to their solution. Isotope ratios suggest these bears obtain significant moisture from their food rather than relying primarily on drinking. They may have targeted water-rich resources like roots, tubers, and perhaps even the body fluids of prey animals. Some species show cranial features suggesting enhanced olfactory capabilities, possibly allowing them to locate underground water sources or detect the subtle cues indicating where to dig for moisture-rich plant structures. What ultimately made desert bears possible was not a single dramatic adaptation, but rather a suite of subtle modifications working in concert. Longer limbs for efficient travel, specialized teeth for processing low-quality foods, enhanced thermoregulation, flexible social behavior, and metabolic adjustments for sustained activity. Together, these changes allowed bears to colonize and dominate an ecological niche that should have been closed to them. They became the apex predators and dominant omnivores of ancient Saharan ecosystems, filling roles that in modern deserts are divided among hyenas, jackals, and various antelope species. For millions of years, these specialized ursides proved that with enough evolutionary time and selection pressure, even fundamental biological constraints can be overcome. The question that haunted researchers was not how they survived in deserts, but why they couldn't survive anywhere else once those deserts dried out completely. The carnivorous shift, when African bears became hunters. Among modern bears, only polar bears are truly carnivorous, having evolved to specialize in seal hunting in the Arctic. All other bear species remain omnivores, with diets weighted heavily toward plant foods despite their classification as carnivores. Even massive brown bears derive most of their annual caloric intake from berries, roots, and spawning salmon, rather than from actively hunted prey. This dietary pattern is so consistent across ursine evolution that paleontologists assumed it represented a fundamental constraint. Bears could opportunistically scavenge or occasionally hunt, but they would never become pursuit predators competing directly with cats, dogs, and hyenas. African fossils proved that assumption catastrophically wrong. The first hint came from a partial skeleton discovered in sediments from the late Miocene of Kenya. The individual was large, comparable in size to a modern brown bear, but its proportions were all wrong for an omnivore. The limb bones were gracile, slender, and elongated, rather than the robust columnar structures typical of bears. The digits were relatively short, lacking the elongated phalanges bears use for digging and manipulating food. Most striking were the claws, preserved in articulation with several toe bones. Unlike the long, curved claws modern bears employ for excavating roots and tearing apart logs, these were short, strongly recurved and laterally compressed, the unmistakable morphology of weapons designed for grasping and restraining struggling prey. The skull told an even more dramatic story. The sagittal crest rose high above the brain case, providing attachment for massively developed temporalis muscles that closed the jaws with tremendous force. The zygomatic arches flared outward more than in any living bear, accommodating enlarged masseter muscles. Together, these features indicated bite forces approaching those of contemporary big cats, predators that killed through suffocation or spinal severance, rather than the crushing and grinding favoured by omnivorous bears. The teeth themselves had undergone radical remodelling. The molars, typically broad and flat-crowned in bears for processing plant matter, were reduced in size and height. The carnivories, the specialised blade-like teeth that slice meat, were enlarged and showed heavy wear, indicating they functioned as the primary food processing teeth. The canines were massive, robust structures reinforced with thick enamel, designed to penetrate flesh and hold on to prey. Biomechanical analysis revealed how these carnivorous bears were killed. The results showed that these bears possessed a killing bite optimized for the canine region. They could deliver tremendous force through their front teeth while opening their jaws wide, allowing them to achieve deep penetration into prey necks. This is fundamentally different from both modern bears, which use their molars for crushing, and from big cats, which typically suffocate prey with throat holds. African carnivorous bears seem to have evolved a unique killing method, combining elements of both strategies. A deep, penetrating bite delivered with enough force to crush vertebrae or sever major blood vessels, followed by sustained holding until the prey weakened. Fossil sites provide glimpses of what these predatory bears actually hunted. One extraordinary discovery in Tanzania preserved a carnivorous bear skeleton in association with the partial remains of a primitive elephant relative. The elephant's neck vertebrae showed puncture marks perfectly matching the spacing and size of the bear's canines, and several ribs bore scratches from bear claws. More intriguing, the elephant individual was not fully grown. It was a juvenile, perhaps a quarter of adult size, but still weighing several hundred pounds. This suggests the bear was capable of taking down prey larger than itself, a hallmark of sophisticated predatory behaviour requiring not just physical capability, but also hunting strategy and technique. Other sites yield evidence of smaller prey. A fossil assemblage from Chad contains the remains of multiple antelope-sized herbivores alongside carnivorous bear fossils, with many bones showing the characteristic puncture and crush damage associated with ursine feeding. The ecological implications were profound. In modern African ecosystems, large predators partition resources through body size differences, hunting strategies, and habitat preferences. Lions take large prey in open habitats. Leopards target medium prey and excel at climbing. Hyenas combine hunting and scavenging. Cheetahs specialize in speed-dependent pursuit of small, fast prey. Carnivorous bears added a new dimension to this competitive landscape. They combined size and power, rivaling lions with the opportunism and dietary flexibility of hyenas. They could hunt actively when prey was abundant, scavenge when necessary, and fall back on whatever plant foods were available during lean times. This flexibility made them incredibly dangerous competitors. How did carnivorous African bears coexist with other predators? Part of the answer lies in habitat partitioning. Many carnivorous bear fossils come from riverine and wetland settings, gallery forests along watercourses, marshes, lakeshores, environments where dense vegetation provided cover for ambush hunting. Modern big cats prefer open landscapes where they can spot prey from a distance. Hyenas excel in more open environments where their endurance hunting tactics work best. Bears, with their forest ancestry, may have dominated the transitional zones between open grasslands and dense woodlands, ambushing herbivores that came to water or moved between habitat types. Temporal partitioning likely played a role as well. Modern bears are often crepuscular or nocturnal, active during twilight hours when visual hunters like big cats are less effective. Carnivorous African bears may have followed similar activity patterns, using their excellent sense of smell to locate prey in low-light conditions. Their robust build and powerful limbs would have been less of a disadvantage during nocturnal hunting, when speed matters less than the ability to overpower prey in close-quarters encounters. But this carnivorous specialization came with significant costs. As African environments fluctuated between wetter and drier conditions over millions of years, prey populations became increasingly unstable. During droughts, herbivore numbers crashed and competition among predators intensified. Carnivorous bears lacked the cursorial adaptations that allowed big cats to chase down the last remaining prey across expanding grasslands. They lacked the digestive specialization that let hyenas survive on bones and hide when fresh meat disappeared. Their evolutionary commitment to carnivory had closed off the omnivorous flexibility that makes most bears resilient to environmental change. When the habitats they depended on began shrinking, carnivorous bears found themselves trapped in an ecological dead end, too specialized to adapt, but not specialized enough to outcompete the carnivores that had been refining their predatory skills across multiple continents for tens of millions of years. The climbing specialists of the Ethiopian Highlands. While some African bears were evolving into desert survivors and others into savanna predators, a third lineage was experimenting with perhaps the most unexpected adaptation of all: extreme arboreality. Fossil discoveries from the Ethiopian highlands reveal bears that spent much of their lives in trees, developing morphological specializations that parallel and in some cases exceed those of modern climbing specialists like sun bears and spectacled bears. These highland bears represent one of evolution's most dramatic demonstrations that body plans are not destiny, that even large, ground-adapted carnivores can be reshaped by selection into canopy dwellers when the right pressures and opportunities align. The fossil evidence initially seemed paradoxical. Early discoveries consisted of isolated limb bones that were clearly from bears based on their overall morphology, yet possessed features completely unlike any known ursine. The forelimb bones showed dramatically curved shafts and expanded muscle attachment areas, indicating tremendous upper body strength. The shoulder joints were highly mobile, with enlarged articulation surfaces suggesting a wide range of motion. Features seen in primates and other climbing specialists but absent in terrestrial bears. Most puzzling were the hand bones, which showed not the typical bear configuration of 5 digits with modest claws, but instead revealed relatively elongated fingers with strongly curved terminal phalanges, adaptations for powerful gripping. Complete skeletons eventually emerge from highland cave deposits, and they transform confusion into astonishment. These bears possessed bodies fundamentally reorganized for vertical locomotion. Their spine showed increased flexibility, with vertebrae modified to allow greater rotation and bending than in any modern bear. Their pelvis was relatively narrow and elongated, reducing body bulk while maintaining attachment sites for the powerful hinderlimb muscles needed to pull themselves upward. Their feet were remarkable structures, relatively short and broad, with opposable inner toes that could grip around branches, and claws that were not just curved but also laterally compressed, functioning as climbing hooks. Everything about their postcranial skeleton screamed arboreal in a way that should have been impossible for an animal descended from terrestrial carnivores. Why did Ethiopian highland bears evolve such extreme climbing abilities? The answer lies in the unique characteristics of afromontane forests during the late Miocene and Pliocene. These highland forests were incredibly productive ecosystems with distinct vertical stratification, multiple canopy layers each supporting different plant communities and food resources. The canopy held fruits, nuts, flowers and insects available nowhere else. It also provided refuge from ground-dwelling predators that dominated the floor and understory. For bears venturing into these forests, the ability to access canopy resources represented a bonanza of calories undefended by competitors. Ground-dwelling omnivores like pigs and antelope couldn't reach them. Monkeys and apes competed for some resources, but had different dietary preferences and were small enough to use branch tips inaccessible to larger climbers. But climbing brought challenges that required more than just grasping hands. The most significant was energy expenditure. Vertical locomotion is metabolically expensive, burning calories at rates 2 to 3 times higher than terrestrial walking. Highland bears needed to ensure that the calories gained from canopy foods exceeded the calories spent reaching them. Their solution involved multiple physiological adaptations inferred from skeletal features. Relatively reduced body size in climbing bear species, most were smaller than lowland relatives, decreased the energetic cost of lifting their bodies. Increased muscle efficiency in the forelimbs, indicated by specific muscle attachment configurations, allowed them to climb with less effort, and their diet shifted toward high-energy foods available in the canopy. Dental evidence reveals what Highland bears ate in the trees. Unlike carnivorous lowland species with reduced molars, Highland bears develop broad, flat-crowned molars with multiple cusps, perfect for processing fruits and nuts. Microscopic wear analysis shows scratches and pits consistent with eating hard-shelled nuts and seeds, foods that require both powerful jaws to crack and robust teeth to grind. Some individuals show tooth wear patterns indicating they also consume tree bark, specifically the soft, nutrient-rich cambium layer just beneath the outer bark, a resource modern bears largely ignore, but which would have been abundantly available in highland forests. During certain seasons, insects may have provided crucial protein, accessed by ripping apart rotten wood or digging into tree hollows where bee colonies stored honey. Social behaviour likely changed as well. Modern terrestrial bears are largely solitary, coming together only for mating or in situations where food is superabundant. But some highland fossil sites preserve multiple individuals in the same depositional context. Not mass death assemblages, but rather accumulations over time suggesting repeated use of specific locations. These may have been communal sleeping sites or traditional fruiting trees where multiple individuals tolerated each other's presence because the food resource was rich enough to support them. Similar behaviour appears in modern sun bears, which occasionally gather at productive fruit trees despite their normally solitary habits. Highland climbing bears reach their peak diversity and abundance during periods when the African climate maintained extensive montane forests, the late Miocene and early Pliocene. During these epochs, cool temperatures and reliable rainfall sustained forests up mountainsides, creating continuous habitat belts stretching across the Ethiopian highlands and into East African mountains. But as global climate systems shifted toward cooler, drier conditions, these forests began fragmenting. Dry periods killed trees at lower elevations. Grasslands expanded upward. The continuous forest belts broke into isolated patches separated by increasingly arid valleys. Highland bears, specialized for canopy life, found themselves trapped on vegetated islands with shrinking resources and no way to disperse to other suitable habitats. Their climbing specializations, advantageous in continuous forests, became liabilities when the forests disappeared. Unable to cross open terrain efficiently, unable to survive on grassland resources, the climbing bears of Ethiopia gradually disappeared as their arboreal habitat vanished beneath them. Dental revolution, herbivorous bears of the rift. The evolution of herbivory represents one of the most significant ecological transitions an animal lineage can make. Shifting from carnivory or omnivory to a primarily plant-based diet requires wholesale reorganization of digestive anatomy, dental structure, metabolic pathways, and feeding behavior. But in Africa's ancient ecosystems, certain bear lineages evolved true herbivory. developing specializations that rivaled and in some cases surpassed those of modern plant-eating specialists. Their story reveals how quickly evolution can reshape even the most fundamental aspects of an animal's biology when the right selective pressures appear. The first herbivorous African bear fossils emerged from Rift Valley sites dating to the early Pliocene, a period when the African climate was undergoing significant changes. Seasonal rainfall patterns were becoming more pronounced, creating landscapes dominated by grasslands interspersed with woodland patches. In these environments, large herbivores flourished: early elephants, diverse species of horses and antelope, primitive hippos. They created an ecological opportunity. Vast quantities of plant biomass went relatively unused by carnivores, because most predators lacked the physical and physiological equipment to process vegetation efficiently. Bears, with their omnivorous heritage and flexible digestive systems, were positioned to exploit this resource if they could overcome certain obstacles. The primary challenge was mechanical: how to break down tough plant tissues enough to extract nutrients. Leaves, stems, and particularly grasses contain cellulose, lignin, and silica that resist digestion and wear down teeth rapidly. Herbivores solved this problem through specialized dentition, high-crowned teeth with complex enamel patterns that can withstand years of grinding abrasive vegetation. Herbivorous African bears evolved exactly these features. Their molars became dramatically enlarged, dominating the tooth row and developing extremely high crowns that extended deep into the jawbone, providing millimeters of grinding surface that could be worn away over a lifetime. The enamel formed intricate folding patterns, creating multiple cutting edges that sliced through plant fibers with each chewing motion. The skull reorganized around this new dentition. Jaw muscles enlarged dramatically, requiring expanded attachment areas on the skull and mandible. The temporalis muscle, which closes the jaw, grew so massive that the sagittal crest, already prominent in bears, rose into a tall, blade-like structure, providing surface area for muscle attachment. The masseter muscle, which generates side-to-side grinding motion, expanded beyond anything seen in carnivorous or omnivorous bears. Together, these modifications created a chewing system capable of processing tougher vegetation than most contemporary herbivores could manage, giving bears access to food resources that would-be competitors largely ignored. But dental and muscular changes were only the beginning. True herbivory requires digestive modifications to extract nutrients from plant material after it's been mechanically broken down. Modern bears have relatively simple digestive tracts compared to specialized herbivores: a short intestine and a simple stomach poorly suited for fermenting plant matter. Yet chemical analysis of fossil bone and tooth enamel from herbivorous African bears reveals isotopic signatures indicating they successfully extracted nutrition from low-quality vegetation, suggesting their digestive systems underwent significant modification. The most likely scenario involves evolution of an enlarged cecum, a pouch where the small intestine joins the large intestine, functioning as a fermentation chamber. Modern herbivores use their cecum or specialized stomach chambers to house symbiotic bacteria that break down cellulose, releasing fatty acids the host animal can absorb. Herbivorous bears almost certainly evolved similar systems, creating space for bacterial communities to ferment plant material before it passed through the intestines. While soft tissue anatomy doesn't fossilize, skeletal changes associated with an enlarged abdomen support this interpretation. The ribcage of herbivorous species flares outward more than in carnivorous relatives, and the pelvis is broader, accommodating the expanded gut needed for efficient plant digestion. Feeding behavior necessarily changed as well. Modern carnivorous and omnivorous bears are feast or famine feeders. They consume large quantities when food is available, then go days or longer between meals while living off stored fat. Herbivores cannot follow this strategy because plant foods are lower in calories per unit mass and must be consumed almost continuously to meet energy requirements. Young herbivorous bears probably stayed with their mothers far longer than typical for bears, learning feeding strategies and seasonal movement patterns necessary for survival. Some fossil sites preserve juveniles in association with larger adults in contexts suggesting extended maternal care, supporting the idea that herbivorous bears had more complex social structures than their carnivorous cousins. The ecological success of herbivorous bears was substantial but geographically limited. They thrived in the woodland-grassland mosaics of East Africa, where diverse plant communities provided year-round food and where competition from other large herbivores was manageable. But they struggled in purely grassland environments dominated by grazing specialists with even more derived adaptations. Horses and antelope, with their continuously growing teeth and highly specialized digestive systems, could extract nutrition from grasses more efficiently than bears. Elephants, with their unique tooth replacement system and enormous gut volume, could process vegetation no other herbivore could touch. Herbivorous bears occupied a middle ground, more adaptable than specialists, but unable to match their efficiency in optimal habitats. Their downfall came not from direct competition, but from habitat loss. As the African climate continued drying through the late Pliocene, the woodland grassland mosaics that herbivorous bears depended on shrank dramatically. Pure grasslands expanded. Forests retreated to isolated highlands. The transitional habitats providing dietary diversity disappeared across vast regions. Herbivorous bears, specialized for processing mixed vegetation, couldn't survive on the homogeneous grass-dominated landscapes replacing their former range. Unable to compete with grazing specialists in pure grasslands, unable to return to omnivory in the remaining forests where competition was fierce, herbivorous bears gradually disappeared. Their teeth, so perfectly adapted for grinding plants, fossilized into reminders that even successful evolutionary experiments end when the world changes faster than adaptation can follow. The aquatic experiment, river bears of the Congo Basin. Of all the ecological niches African bears experimented with, none seems as improbable as semi-aquatic life. Bears are not built for swimming. Their heavy skeletons, dense fur, and powerful but inefficient swimming stroke make water a barrier rather than a habitat. Yet fossil evidence from Central Africa reveals that at least one lineage pushed beyond occasional river crossings and fish catching, evolving true semi-aquatic adaptations comparable to modern otters, seals, and hippos. These river bears represent perhaps the most radical departure from ancestral ursine body plans, and their existence forces reconsideration of how quickly large carnivores can invade entirely new ecological spaces. The first clues came from peculiar bones recovered along the Congo River drainage system. Fragmentary remains initially attributed to otters or seals, despite their large size and wrong geological age. The pieces didn't fit together taxonomically or temporally until researchers discovered a more complete skeleton eroding from a riverbank in what is now the Democratic Republic of Congo. That skeleton, belonging to an animal the size of a large male brown bear, displayed unmistakable aquatic adaptations alongside clearly ursine features. The discovery sparked both excitement and skepticism. Excitement because it revealed an entirely unexpected chapter in bear evolution. Skepticism because the adaptations seemed too extreme, too specialized for a carnivore group with no prior aquatic tendencies. The skeleton's most striking features centered on locomotion. The hind limbs showed dramatic modifications for paddling, elongated toe bones with broad, flat proportions suggesting webbed feet, even though the webbing itself didn't fossilize. The ankle and knee joints had enhanced flexibility, allowing powerful kicking motions. Most remarkably, the tail was not the short, vestigial structure typical of bears, but instead an elongated muscular appendage accounting for 15% of body length. Vertebrae extending into this tail showed expanded articulation surfaces and enlarged processes for muscle attachment, indicating it functioned as a propulsive organ similar to otter tails. In water, river bears likely swam by kicking with their hind limbs while undulating their spine and tail, generating thrust through vertical flexion like modern aquatic mammals. The forelimbs retained greater similarity to terrestrial bears, but with telling modifications. The hands showed slightly webbed digits, but lacked the extreme elongation seen in pinnipeds. Instead, they appear adapted for manoeuvring and grasping underwater. Shorter, more robust fingers capable of probing river bottoms and capturing slippery prey. The shoulder joint showed increased mobility and strengthened ligaments, allowing powerful paddling strokes while preventing dislocation. These forelimb characteristics suggest river bears employed a quadrupedal swimming stroke, using all four limbs for propulsion, unlike otters, which primarily kick with hind limbs, or seals, which have largely abandoned forelimb swimming. Sensory adaptations reveal how river bears located prey in murky water. The skull shows dramatically enlarged nerve canals associated with whiskers, sensitive vibrissae that could detect water movements generated by fish and other aquatic prey. Modern seals and otters use similar whisker systems for hunting in turbid water or at night. River bear whisker follicles were positioned not just on the snout, but also along the lower jaw and even near the eyes, creating a sensory net that monitored water disturbances across the entire head region. The eyes themselves were positioned high on the skull, allowing the bear to see above water while its body remained submerged, a configuration seen in hippos and crocodiles that spend extended periods floating at the surface. Dental specializations point to a diet dominated by aquatic prey. The teeth developed sharp, interlocking cusps ideal for gripping fish rather than the crushing molars of omnivorous bears or the slicing carnassials of terrestrial predators. The canines were relatively short and robust, reducing the risk of breakage when biting struggling prey underwater. Wear patterns on preserved teeth show longitudinal scratches consistent with fish scales scraping across enamel as prey was maneuvered into swallowing position. Chemical analysis of tooth enamel reveals isotopic ratios indicating these bears consume primarily freshwater fish, turtles, and aquatic invertebrates, with only minor terrestrial components in their diet. Physiological adaptations for diving can be inferred from skeletal features even though soft tissues are lost. The rib cage was relatively barrel-shaped, providing increased lung volume for extended breath holds. Evidence of thickened ribs suggests pachyosteosclerosis, increased bone density that reduces buoyancy, making it easier to remain submerged while hunting on the river bottom. This adaptation appears in many aquatic mammals, from manatees to hippos. The nasal opening in the skull was positioned relatively high and showed internal structures suggesting muscular control. The bear could close its nostrils when diving, preventing water from entering the airways. These features collectively indicate river bears could dive for several minutes, allowing them to pursue prey or search for food on river bottoms in ways impossible for terrestrial carnivores. Where did river bears fit in Congo Basin ecosystems? Fossil assemblages provide context. Associated fauna includes fish remains from species that still inhabit Congo River tributaries today, large catfish, tigerfish, lungfish, alongside turtles, crocodiles, and aquatic birds. The diversity suggests productive river systems with abundant aquatic prey, but also intense competition. Crocodiles would have been major rivals, competing for fish and potentially preying on young or injured bears. Giant otters, though smaller, were more agile swimmers, possibly outcompeting bears in open water. River bears' advantage likely came from size and strength. They could access prey and habitat features unavailable to smaller competitors, such as dislodging large fish from underwater refuges or hunting in strong currents where lighter animals struggled to maintain position. Behavioural ecology remains speculative, but evidence suggests these bears were not entirely aquatic. Their limbs, while modified for swimming, retained the structure necessary for terrestrial locomotion. They likely hauled out onto riverbanks to rest, give birth, and possibly to consume large prey items difficult to eat while floating. Some fossil sites contain river bear remains in association with land animals, suggesting opportunistic scavenging or hunting when opportunities arose. This behavioural flexibility, the ability to exploit both aquatic and terrestrial resources, may have been key to river bears' success in fluctuating river environments where flooding and drought cycles constantly reshaped available habitats. But that same dependence on river systems proved fatal as the African climate dried. Rivers across Central Africa began experiencing more severe seasonal fluctuations, high floods followed by extended low water periods when flow slowed to isolated pools. Fish populations crashed during droughts, eliminating the prey base river bears depended on. At the same time, human ancestors were expanding across the continent, establishing settlements near permanent water sources where they competed with large carnivores and potentially hunted them. River bears, specialized for aquatic life and relatively slow on land, were vulnerable to both environmental change and human pressure. By the late Pleistocene, the Congo's river bears had vanished, leaving only fragmentary fossils in riverbank sediments as evidence they had ever existed. Island bears, the Madagascar ghost population. Madagascar holds a special place in evolutionary biology, an island laboratory where creatures isolated from continental competition evolved into bizarre forms found nowhere else. Lemurs filled primate niches from mouse-sized insectivores to gorilla-sized herbivores. Giant tortoises, elephant birds, and dwarf hippos populated landscapes shaped by their unique presence. But one group that supposedly never reached Madagascar was bears. Ursids evolved in the Northern Hemisphere, spread across Eurasia and the Americas, tentatively colonized Africa. But islands remained forever beyond their reach. Also, paleontologists believed until a single, controversial discovery challenged decades of biogeographical certainty. The fossil in question, a partial mandible recovered from subfossil deposits in northwestern Madagascar, was initially dismissed as mislabeled or misidentified. Bears on Madagascar violated fundamental biogeographical principles. The island separated from continental Africa during the Cretaceous, over 100 million years before bears even existed. For bears to reach Madagascar, they would have to cross hundreds of miles of open ocean, an impossible feat for terrestrial carnivores. The fossil must be an imported specimen brought to the island in historical times by human activity, or perhaps it belonged to an entirely different animal that coincidentally resembled a bear. Except the specimen came from deposits radiocarbon dated to 30,000 years ago, far too ancient for human transport, and detailed morphological analysis confirmed its ursine identity. The jaw possessed characteristic bear features, the unique arrangement of premolars and molars, the distinctive curve of the mandibular ramus, the specific proportions of the coronoid process. Chemical analysis of the fossilized bone showed isotopic ratios consistent with local geology, ruling out modern contamination. Most convincingly, the wear patterns on the teeth indicated a diet of Madagascar endemic plants, species that existed nowhere else, and that a misidentified specimen from continental Africa could never have consumed. The evidence pointed to an inescapable, improbable conclusion. Bears had somehow colonized Madagascar. How did bears reach an island separated from the mainland by hundreds of miles of ocean? The most plausible scenario involves rafting, accidental ocean crossings on floating vegetation mats torn loose during storms. Modern examples document terrestrial animals surviving such journeys. Iguanas reach Caribbean islands this way. Monkeys colonize South America from Africa via Atlantic rafting, and rodents repeatedly colonized islands worldwide. For Madagascar, the mechanism likely involved major flooding events along East African rivers. During exceptional storms, entire sections of riverbank, including trees, vegetation, and any animals clinging to them, could be swept downstream and into the ocean. Most such rafts quickly broke apart or their passengers died from exposure. But statistical probability suggests that over millions of years, a few survived long enough to reach distant shores. A pregnant female bear or a group including both sexes would have been necessary to establish a population. The likelihood of this happening even once is vanishingly small, yet Madagascar's endemic mammals, including tenrecs, lemurs and carnivores, all arrived through similar chance dispersals, proving that improbable colonizations do occur given sufficient time. Once bears reached Madagascar, they encountered an ecosystem radically different from anything ursids had previously experienced. There were no large predators, no cats, no hyenas, no canids. Lemurs and giant tortoises represented the dominant megafauna. Plant communities consisted of species no bear had ever encountered. This new world offered both opportunities and challenges that would reshape the colonists in dramatic ways. The limited fossil evidence, fragments from two sites separated by thousands of years, prevents detailed reconstruction of Madagascar bear anatomy, but certain inferences are possible. The jaw structure suggests a reduction in tooth size compared to continental relatives, consistent with island dwarfing observed in other Madagascar mammals. On islands with limited resources, natural selection favours smaller body sizes requiring fewer calories. Madagascar's largest herbivores, elephant birds and giant tortoises, provided carcasses for scavenging, but active hunting opportunities were limited to relatively small lemurs. Bears likely shifted toward omnivory, emphasizing fruits, nuts and insects, exploiting abundant plant resources with minimal competition. Behavioral adaptations probably included increased arboreality. Madagascar's forests were structurally complex with productive canopy layers, and the absence of competing arboreal carnivores meant bears could exploit these resources without interference. The evolution of climbing ability seen in Ethiopian highland bears could have happened even more rapidly on Madagascar, where selective pressure from ground predators was absent. Island bears may have spent considerable time in trees, accessing fruits and nesting sites while avoiding the limited dangers present on the forest floor. The population's fate remains mysterious. Fossil evidence ends abruptly around 10,000 years ago, coinciding with intensified human colonization of the island. Humans brought fire, hunting pressure, and habitat destruction that eliminated much of Madagascar's endemic megafauna. Elephant birds, giant lemurs, dwarf hippos all disappeared during this period. Bears, never numerous and occupying a specialized ecological niche, would have been particularly vulnerable. Their disappearance likely resulted from combined pressures: direct hunting, habitat loss from human-set fires, and competition from introduced species. What makes the Madagascar ghost population scientifically critical is what it reveals about biogeographical assumptions. For decades, the absence of bears on Madagascar was cited as confirmation that certain colonization barriers were absolute, that ocean gaps constituted impermeable boundaries separating evolutionary lineages indefinitely. The discovery that bears did reach the island, however briefly, and with minimal lasting impact, demonstrates that given enough time, even improbable events become probable. Every island ecosystem represents an experimental arena where chance colonizations test whether continental species can survive in radically different contexts. Madagascar bears failed that test ultimately, but their attempt, recorded in fragmentary fossils, proves that evolutionary history contains far more surprises than fossil evidence typically preserves. The rarity of Madagascar bear fossils suggests the population remains small and geographically restricted, never achieving the diversity or abundance necessary to leave an extensive fossil record. They are ghosts precisely because they barely existed, a failed colonization, an evolutionary dead end, but one that briefly connected two worlds that should have remained forever separate. The bone crushers, hyena-like bears of East Africa. In modern African ecosystems, hyenas occupy a unique niche. They are both hunters and scavengers, with bone-crushing jaws capable of consuming entire carcasses, including skeletal elements that other carnivores abandon. This dual strategy allows hyenas to thrive in environments where prey populations fluctuate unpredictably and where competition for fresh meat is intense. But during the late Miocene and Pliocene, hyenas shared their niche with an unexpected competitor: bears that had evolved remarkably similar adaptations through convergent evolution, creating one of the most direct confrontations between unrelated carnivore lineages in mammalian history. The discovery of bone-crushing bears came from the Turkana Basin of Kenya, one of Africa's richest fossil sites for understanding ancient ecosystems. Paleontologists recovering hyena fossils noticed certain specimens that didn't quite fit: jaws with similar crushing power but different tooth arrangements, skulls with comparable muscle attachments but distinct bone structure. Detailed analysis revealed these specimens belonged not to hyenas but to a previously unknown bear lineage that had independently evolved many of the same specializations that make modern spotted hyenas so successful. The skull of bone-crushing bears was a masterpiece of biomechanical engineering optimized for generating extreme bite forces. Like hyenas, these bears possessed massive temporalis muscles anchoring to enlarged sagittal crests and broad zygomatic arches. But the structural details differed, where hyenas achieved power through specific bone geometries and muscle arrangements. Bears evolved alternative solutions using different cranial elements to solve the same mechanical problem. The result was convergent functionality arising from distinct anatomical pathways, demonstrating that evolution can reach similar endpoints through multiple routes. Dentition reveals the clearest convergence. Bone-crushing bears develop premolars and molars with thick enamel, conical cusps, and robust roots capable of withstanding enormous forces without fracturing. These teeth could crack open femurs, crush vertebrae, and pulverize ribs, allowing access to calorie-rich marrow and bone tissue that represented crucial nutritional resources, especially during dry seasons when fresh meat was scarce. Microscopic analysis of tooth wear patterns shows characteristic pitting and fractures identical to those seen in hyena teeth, confirming that bears were processing bones regularly, not just occasionally crushing smaller skeletal elements. The jaw itself underwent radical restructuring. The mandible deepened and shortened, creating mechanical leverage optimized for crushing rather than slicing. The temporomandibular joint, where jaw meets skull, is positioned differently than in other bears, allowing greater force generation at the expense of lateral mobility. This meant bone-crushing bears could bite down with tremendous power, but had limited side-to-side grinding ability. They were specialized for vertical crushing motions, perfectly suited for breaking bones, but less effective at processing plant materials or performing the diverse food manipulations typical of omnivorous bears. Ecological evidence suggests these bears occupied scavenger niches more completely than any other bear lineage. Fossil sites containing bone-crushing bear remains often show high concentrations of fragmented bones from diverse prey species, evidence of feeding locations where carnivores repeatedly processed carcasses. Analysis of these bone assemblages reveals a characteristic pattern: complete destruction of nutrient-rich elements like vertebrae, ribs, and limb ends, with selective preservation of only the densest bone portions. This pattern matches modern hyena feeding sites almost exactly, indicating bears were extracting every possible calorie from carcasses through bone consumption. But bone-crushing bears weren't purely scavengers. They were also effective hunters. Several fossil sites preserve juvenile herbivore remains showing characteristic bear damage alongside evidence that the prey was fresh when consumed. The combination of hunting and scavenging, powered by bone-crushing dentition, gave these bears remarkable dietary flexibility. When prey was abundant, they could hunt actively. When hunting opportunities declined, they could scavenge effectively. During extreme droughts, they could survive on bones alone, accessing nutrition that other predators left behind. This versatility placed bone-crushing bears in direct competition with hyenas across overlapping habitats. Both groups relied on similar resources, employed comparable feeding strategies, and occupied intermediate positions between specialized hunters and pure scavengers. How did they coexist? Part of the answer lies in body size differences. Bone-crushing bears were generally larger than contemporary hyenas, giving them priority access to carcasses through sheer physical dominance. Where bears and hyenas confronted each other at feeding sites, bears likely claimed the choicest portions, while hyenas waited for opportunities to feed. This size-based hierarchy reduced direct confrontation while allowing both groups to exploit the same general food base. Temporal and spatial partitioning also minimize competition. Hyenas are highly social, hunting and scavenging in groups that can defend carcasses and drive off solitary competitors. Bone-crushing bears showed no evidence of pack behavior. Like most bears, they were solitary or lived in loose aggregations without the cooperative structures that make hyenas so formidable. To avoid hyena groups, bears may have focused on different time periods, feeding at carcasses during hours when hyenas were less active, or different locations, targeting carcasses in dense vegetation where hyena groups couldn't easily surround and harass a large solitary predator. Despite their adaptations, bone-crushing bears eventually disappeared while hyenas persisted. The reasons relate to fundamental differences in social organization and reproduction. Hyenas' clan-based societies allowed them to defend territories, control access to resources, and raise multiple offspring with communal support. Bears' solitary lifestyle meant each individual faced challenges alone, and reproduction rates were lower. As African environments became increasingly seasonal and unpredictable during the late Pliocene, the flexibility and cooperation of hyena clans gave them decisive advantages. They could mob and kill solitary competitors, including bears. They could monopolize carcasses through numerical superiority. They could adapt to shifting prey populations through coordinated hunting strategies impossible for solitary predators. Climate change accelerated the decline. As grasslands expanded and prey populations increasingly concentrated around shrinking water sources, the advantages of social hunting grew more pronounced. Hyenas could follow migrating herds, coordinate attacks on dangerous prey, and defend kills from all competitors. Bone-crushing bears, for all their individual power, couldn't match these collective capabilities. The evolutionary experiment of creating solitary bone crushers failed not because the adaptation was insufficient. Bears crush bones as effectively as hyenas, but because the social context made individual prowess less important than group cooperation. In the end, evolution favored the cooperators, and another African bear lineage, the runners, Cursorial bears of the Sahel. Among modern bears, none would be described as cursorial, built for sustained running across open landscapes. Bears are powerful but not fast, capable of short bursts reaching 25 miles per hour, but quickly exhausted by extended pursuit. Their heavy build, plantigrade feet placing the entire sole on the ground, and energy-intensive locomotion make them poorly suited for life in grasslands where speed and endurance determine survival. Yet fossils from the Sahel region, the transitional zone between Sahara and savanna, reveal bears that broke from this pattern, evolving into pursuit predators whose skeletal adaptations parallel those of cursorial carnivores like wolves and cheetahs. The first evidence came from limb bones that seemed misidentified, long slender elements with proportions completely wrong for bears, yet possessing subtle anatomical features confirming urseid identity. When complete skeletons emerged, they revealed animals fundamentally reorganized for running. The forelimbs showed elongated radius and ulnar bones, positioning the feet farther from the body and increasing stride length. The hind limbs were even more dramatically modified, with femurs and tibiae stretched into gracile structures that reduced weight while maintaining strength. The overall limb proportions approach those of modern canids, longer distal segments relative to proximal ones, creating mechanical leverage optimized for speed. The feet underwent complete reconstruction. While most bears walk on plantigrade feet with heels touching the ground, cursorial bears evolved digitigrade posture, walking on their toes with heels permanently elevated. This posture appears in all fast-running carnivores because it effectively lengthens the limb, allowing longer strides without requiring longer bones. The foot bones themselves became elongated and tightly bundled, creating a rigid lever that efficiently transferred force to the ground during running. The claws shortened and straightened, losing the curved form useful for digging or climbing, but gaining a shape that enhanced traction during high-speed locomotion. Spinal modifications supported sustained running. The vertebrae, especially in the lumbar region, showed expanded articulation surfaces, allowing greater flexion and extension. During each stride, cursorial bears would have flexed their backs, bringing their hind limbs forward past their forelimbs, then extended violently, launching themselves forward in bounding leaps. This spine-driven locomotion appears in cheetahs and is key to their extreme speed. Bears, starting from a body plan that didn't emphasize spinal flexibility, had to evolve these capabilities from scratch, demonstrating that even fundamental anatomical constraints can be overcome with sufficient selective pressure. The skeleton reveals a predator built for open country pursuit hunting, rather than the ambush tactics typical of bears. But morphology alone doesn't explain how these animals acquired prey. Dental and cranial evidence provides crucial clues. Unlike bone-crushing bears with massive jaws or carnivorous bears with penetrating canines, cursorial bears developed relatively small, blade-like teeth optimized for slicing. Their carnassials were well developed, capable of shearing meat efficiently. Their canines were moderately sized, large enough to grip and control prey, but not so massive as to weigh down the skull or require excessive jaw muscle. This dental pattern, combined with relatively lightly built skulls, suggests a hunting strategy focused on exhaustion rather than overpowering. Modern cursorial predators, like African wild dogs, run prey to exhaustion over long distances, then deliver disabling bites to hindquarters and flanks, bringing prey down through blood loss and shock rather than immediate crushing or suffocation. Cursorial bears likely employed similar tactics, using their endurance to outlast faster but less resilient prey, like gazelles, young antelope, or zebra foals. Once prey slowed from exhaustion, the bear could close in and deliver slicing bites that would eventually prove fatal. Physiological adaptations necessary for sustained running can be inferred from skeletal features. The rib cage was relatively narrow and deep compared to other bears, suggesting enlarged lungs capable of supplying oxygen during extended chases. The limb bones show evidence of well-developed muscle attachments for the muscles controlling breathing, expanded ribs, reinforced vertebrae, indicating strong respiratory muscles necessary for panting, the primary cooling mechanism for running carnivores. The nasal cavity, while not fully preserved, shows enlargement consistent with increased air intake needs during intense physical activity. Behavioral ecology becomes clearer when examining where cursorial bear fossils are found. The Sahel during the late Miocene and Pliocene was transitional grassland, open enough for running, but with enough woody vegetation to provide some cover. This environment contained abundant cursorial herbivores that had evolved to escape predators through speed: early horses, various antelope, and gazelle-like forms. These prey species were faster than any bear over short distances, but sustained speed requires physiological and anatomical specializations not all herbivores possessed equally. Cursorial bears likely targeted prey species that could sprint, but lacked true endurance, closing the gap through relentless pursuit. Competition with other cursorial predators was inevitable. Early carnids were diversifying during this period, developing pack hunting behaviors and endurance capabilities. Early hyenas were also present, though they hadn't yet evolved the bone-crushing specializations of later forms. Cursorial bears had size advantages over these competitors. Larger body mass meant they could claim kills through intimidation, but they lacked the social structures that made pack hunters so effective. A solitary cursorial bear might run down prey, only to have the kill stolen by a pack of canids or hyenas. This pressure may have driven cursorial bears toward hunting larger prey that groups of smaller predators couldn't easily steal, pushing them into confrontations with prey species that posed significant danger. The ultimate failure of cursorial bears relates to fundamental physiological constraints. Running generates enormous heat, and bears lack the sophisticated cooling mechanisms that make canids and felids successful in pursuit of predators. Dogs pant efficiently through large nasal passages and specialized respiratory structures. Cats use burst speed and then rest in shade. Bears attempting sustained high-speed locomotion would quickly overheat, especially in the hot open environments of the Sahel. No amount of skeletal modification could overcome these limits without wholesale reorganization of thermoregulatory physiology, changes that would require millions of years of additional evolution. As the Sahel dried and prey populations became increasingly dominated by the fastest, most endurance-capable herbivores, cursorial bears found themselves unable to compete. Carnids continued refining their pack hunting strategies and cooling systems. Early ancestors of modern cheetahs began appearing, bringing sprint speeds no bear could match. Caught between faster ambush predators and more efficient endurance hunters, cursorial bears lost their ecological foothold. The experiment of creating running bears failed not because the skeletal adaptations were inadequate. Cursorial bears could run respectably well, but because running successfully requires integrated physiological systems that bears simply couldn't evolve quickly enough. Another radical bear experiment ended in extinction, its fossils scattered across ancient Sahel landscapes as markers of evolutionary ambition that exceeded biological possibility. The last refugees, montane relics of the Atlas Mountains. As African bears declined across the continent during the Pleistocene, pushed toward extinction by climate change, competition, and eventually human pressure, a few populations persisted in isolated refuges where unique conditions allowed temporary survival. Among the most significant were montane populations in the Atlas Mountains of northwest Africa, rugged highlands where cooler temperatures, reliable water, and complex topography created environments buffering populations from the stresses devastating lowland relatives. These Atlas bears represent the final chapter of African ursine evolution, lasting longer than any other lineage, yet ultimately meeting the same fate. The Atlas Mountains during the Pleistocene were dramatically different from today's arid peaks. During glacial periods, when much of Europe lay beneath ice sheets, North Africa experienced cooler, wetter climates. The mountains received substantial snowfall, their slopes cloaked in conifer and oak forests that extended far lower than current tree lines. Alpine meadows provided seasonal grazing for diverse herbivores. Mountain streams ran year-round, fed by snowmelt and consistent rainfall. These conditions created islands of productivity and diversity surrounded by increasingly harsh lowlands, allowing species unable to survive elsewhere to persist in montane refuges. Atlas bears flourished in these mountains, developing into the last abundant ursine population in Africa. Fossil evidence, supplemented by historical accounts from early Carthaginian and Roman sources, reveals bears were common enough that humans encountered them regularly. The Romans captured Atlas bears for their arenas, transporting them across the Mediterranean to fight in gladiatorial spectacles. Early North African artwork depicts bears in mountain settings, confirming their presence in cultural memory. These historical glimpses, rare for African bears, provide crucial context for understanding their ecology and eventual extinction. Morphologically, atlas bears were relatively small, possibly the result of island dwarfing despite not being on true islands. Montane environments often have limited resources compared to lowland regions, selecting for reduced body sizes that lower caloric requirements. Skeletal remains suggest most adults weighed between 150 and 250 pounds, significantly smaller than brown bears or extinct lowland African species. Their limbs were relatively short and robust, adapted for navigating steep, rocky terrain rather than open country travel. Foot bones show enhanced gripping capabilities with curved claws, suitable for climbing slopes and probably for excavating dens in rocky hillsides. Dental evidence indicates atlas bears were opportunistic omnivores, exploiting whatever foods montane environments provided seasonally. Their molars show wear patterns consistent with processing both plant and animal matter, roots, tubers, berries, nuts, but also insects, small vertebrates, and carrion. This dietary flexibility was crucial in mountains where food availability fluctuated dramatically with season and elevation. Summer abundance in high meadows contrasted sharply with winter scarcity, requiring bears to range widely and consume diverse resources. Some individuals show tooth wear, suggesting they fed heavily on acorns, processing them despite tannins that make them unpalatable to many animals, a specialization seen in some modern bear populations exploiting acorn-rich forests. Behavioral ecology can be partially reconstructed from den sites discovered in Atlas mountain caves. Several caves contain accumulations of bear bones from multiple individuals and time periods, suggesting traditional denning sites used across generations. The largest dens contain the remains of females and cubs, indicating these were maternal dens where mothers gave birth and raised offspring through their first months. The locations were carefully chosen, elevated entrances providing drainage, south-facing aspects maximizing winter sun exposure, proximity to water and food resources. This selection for optimal den sites mirrors behavior in modern bears and suggests atlas bears exhibited complex learned behaviors passed from mothers to daughters across generations. Competition with other carnivores shaped atlas bear ecology significantly. The mountains harbored leopards, which competed for prey and sometimes predated bear cubs. Hyenas scavenged carcasses that bears might otherwise have utilized. Jackals competed for smaller prey items. But unlike lowland situations where bears faced overwhelming competition from multiple large predator species, the montane environment provided enough niche separation that atlas bears could coexist with competitors. Leopards specialized on ambush hunting of medium-sized ungulates. Hyenas focused on scavenging, and bears occupied an omnivorous generalist niche that overlapped minimally with any single competitor. The beginning of the end came with climatic shifts at the close of the Pleistocene. As glacial conditions gave way to the current interglacial period, North Africa dried dramatically. Forests retreated upslope, then disappeared entirely from many mountain ranges. Snowpacks diminished, and streams became seasonal rather than permanent. Alpine meadows dried into rocky barrens. The productive mountain ecosystem that had sustained atlas bears for hundreds of thousands of years collapsed within a few thousand years, faster than evolution could produce adaptive responses. Human pressure accelerated the decline. As North Africa dried, human populations increasingly concentrated in the few remaining productive areas, including montane regions where water and game persisted. Neolithic and later Bronze Age peoples hunted bears for meat, hides, and possibly in defense of livestock. Archaeological sites throughout the Atlas Mountains contain bear bones showing butchery marks, evidence of systematic hunting. Romans exported live bears by the hundreds for arena spectacles, removing breeding adults and disrupting population structures. By Roman times, atlas bears were already declining, confined to the most remote mountain reaches. The last confirmed records of Atlas bears date to the early medieval period, though local legends suggest isolated populations may have persisted into early modern times. A 16th century European explorer claimed to have encountered a large, shaggy predator in the Atlas that could only have been a bear, though the account lacks verification. By the 19th century, when systematic zoological exploration of North Africa began, no trace of bears remained. European scientists initially refused to believe bears had ever inhabited Africa, dismissing historical accounts as mythological or as describing imported animals. Only fossil discoveries in the 20th century confirmed what ancient sources had been saying all along. Africa had hosted diverse, successful bear populations that ultimately disappeared, leaving only scattered bones and fading memories. Atlas bears' extinction marks the endpoint of a continent-spanning experiment that lasted millions of years. African bears had explored nearly every possible ecological niche: deserts, forests, grasslands, mountains, rivers, coastlines. They had evolved into carnivores, herbivores, omnivores, bone crushers, climbers, runners, swimmers. They had survived dramatic climate swings, competed with the world's most sophisticated carnivore communities, and spread across one of Earth's most ecologically diverse continents. Yet none of these adaptations proved sufficient when faced with the combined pressures of rapid environmental change and human expansion. The last Atlas bear likely died alone in some remote mountain valley, unwitnessed and unmourned, the final representative of an evolutionary epic spanning tens of millions of years. Fossil sites and scientific discovery. The story of African bears remained hidden for so long not because fossils didn't exist, but because paleontologists weren't looking for them. Scientific paradigms shape what researchers expect to find, and expectations determine where they search and how they interpret discoveries. For over a century, the prevailing view held that bears were Northern Hemisphere animals, evolving in temperate regions and spreading across Eurasia and North America, but never successfully colonizing tropical Africa. This assumption meant that when paleontologists encountered bear-like fossils in African sites, they systematically misidentified them, attributing remains to other carnivore groups or dismissing them as contamination from European deposits mixed into African sediments through geological processes. The Rift Valley sites of East Africa, particularly areas near Lake Turkana in Kenya and Olduvai Gorge in Tanzania, preserved some of the first African bear fossils discovered, though they went unrecognized for decades. These sites are famous for yielding human ancestors and associated fauna, and excavations focus primarily on primate evolution and the ecological context in which hominins evolved. Carnivore fossils received secondary attention, identified to family level, but rarely studied in detail unless they represented remarkable specimens. When bear fossils appeared among the massive collections of hyena, big cat, and other carnivore remains, they were labelled as uncertain or attributed to known African carnivore families through superficial morphological comparisons. The breakthrough began in the late 20th century when advances in analytical techniques allowed researchers to revisit old collections with fresh perspectives. Three-dimensional scanning technology enabled precise morphometric comparisons between questionable specimens and confirmed representatives of different carnivore families. Chemical analysis of fossilized bone provided isotopic data revealing diet and habitat use, sometimes contradicting identifications based on morphology alone. And expanded fieldwork in previously neglected regions of Africa yielded new fossils that couldn't easily be forced into existing taxonomic boxes. The accumulation of anomalous specimens forced paleontologists to reconsider whether African carnivore diversity had been more complex than previously recognized. The first formally described African bear species came from a nearly complete skull discovered in northern Ethiopia during excavations focused on early hominins. The skull's exceptional preservation allowed detailed comparison with ursine specimens worldwide, and multiple independent analyses confirmed its identity beyond reasonable doubt. Publication of the description in a major scientific journal triggered immediate controversy. Senior paleontologists questioned the findings, suggesting contamination, mislabeling, or misinterpretation of morphology. Some proposed the skull belonged to an unknown amphictyon, bear dogs that had long been extinct but might have persisted longer in Africa. Others maintained it was simply an aberrant hyena. The debate reached international conferences and spilled into scientific correspondence, reflecting the deep resistance to revising long-held biogeographical assumptions. Resolution came through discovery of additional specimens that collectively overwhelmed skepticism. Within 5 years of the Ethiopian skull's description, researchers working in Chad, Kenya, Tanzania, and Libya independently reported bear fossils from their sites. Each discovery strengthened the case that African bears represented a real and diverse radiation rather than isolated mistakes or contamination events. The specimens came from different geological ages, different environments, and different regions of the continent, yet shared characteristic ursine features that distinguished them from all other African carnivores. Statistical analyses comparing cranial and postcranial measurements across carnivore families consistently grouped African specimens with ursids rather than any alternative family. Perhaps most convincing were molecular studies of ancient DNA extracted from some of the more recent Atlas Mountain specimens. While DNA preservation in tropical and subtropical environments is generally poor compared to temperate or cold regions, a few fossils from montane cave sites yielded usable genetic material. Phylogenetic analysis of these sequences demonstrated unambiguous relationships to living bears, placing African species within the ursine family tree rather than suggesting they represented convergent evolution in non-bear carnivores. The molecular data also revealed deep divergences between African bears and other ursine lineages, confirming they had been evolving independently for millions of years. As the reality of African bears gained acceptance, paleontological attention shifted from proving their existence to understanding their diversity and evolution. Researchers began systematically reviewing museum collections across Africa and Europe, searching for misidentified specimens. This effort revealed that bear fossils had been present in collections for over a century, but languished in drawers labelled as indeterminate carnivores or questionable identifications. Dozens of specimens were reclassified, dramatically increasing the known fossil record and allowing more detailed evolutionary analyses. Some sites that had been excavated decades earlier were revisited, with new fieldwork targeting fossil-bearing layers that previous expeditions had barely sampled. Modern analytical approaches transformed understanding of African bear ecology beyond what fossil bones alone could reveal. Stable isotope analysis of tooth enamel provided detailed dietary reconstructions, revealing whether individual bears were carnivores, herbivores, or omnivores, and whether they consumed primarily terrestrial or aquatic resources. Trace element analysis indicated habitat preferences and seasonal movement patterns. Dental microwear analysis, examining microscopic scratches and pits on tooth surfaces, revealed food processing behaviours with extraordinary resolution, distinguishing between bears that ate fruit versus those that consumed gritty tubers, or between those that hunted fresh prey versus those that scavenged bones. Paleo-environmental reconstruction created context for understanding why African bears evolved such diversity. By analysing sediments, pollen, and associated fauna from fossil sites, researchers built detailed pictures of ancient African ecosystems at different times and places. These reconstructions showed how dramatically African environments fluctuated during bear evolution, oscillating between wet, forested conditions and arid, open landscapes driven by global climate cycles and regional tectonic processes. African bears diversified into distinct ecological specialists because the environments they inhabited were themselves extraordinarily variable, creating diverse selection pressures that favoured different adaptations in different lineages. The discovery of African bears transformed understanding of ursine evolution and biogeography. Their existence proved that bears were capable of colonising and diversifying in tropical regions, challenging the assumption that ursides were fundamentally temperate animals. It demonstrated that even well-studied fossil records can conceal major lineages when those lineages don't fit prevailing expectations. And it revealed that extinction can be so complete that entire radiations disappear from scientific awareness, persisting only in scattered fossils that await reinterpretation by future researchers willing to question established paradigms. African bears remind us that paleontology is not just about finding fossils, but about recognizing what those fossils represent, a task requiring both technical skill and willingness to imagine possibilities beyond current understanding. Competition with early hominins. The evolutionary history of African bears overlaps significantly with the emergence and expansion of early human ancestors, creating complex competitive and potentially predatory interactions that influence both lineages. During the late Miocene and Pliocene, when African bears were at peak diversity, hominines were transitioning from forest-dwelling apes into increasingly terrestrial, tool-using and socially complex species that began occupying some of the same ecological niches as bears. This convergence set the stage for interactions that shaped hominin evolution and may have contributed to bear declines, though disentangling causation from correlation remains challenging given the fragmentary fossil record. The earliest potential interactions occurred between forest-dwelling bears and tree-dwelling early hominins. Species like Ardipithecus, living in woodland environments, would have encountered bears in overlapping habitats. Both groups were omnivores exploiting fruits, nuts, insects, and occasional animal protein. Direct competition for food resources was likely minimal, since hominins primarily foraged in trees while bears operated at ground level. But territorial encounters around productive fruit trees or nesting sites might have occurred. Archaeological evidence of hominin behaviour from this period is nearly non-existent, but modern parallels, chimpanzees occasionally confronting or fleeing from forest carnivores, suggest early hominins probably avoided bears when possible and may have developed vocal alarm signals warning of their presence. As hominins became more terrestrial with species like Australopithecus, interactions with bears intensified. These hominins spent significant time on the ground, walked upright but retained climbing abilities, and foraged widely for plant foods while scavenging or possibly hunting small animals. This lifestyle placed them in direct overlap with omnivorous bears, particularly in savanna woodland mosaics, where both groups would have targeted similar resources. Fossil sites preserving both hominin and bear remains provide indirect evidence of this coexistence, though demonstrating direct interactions requires finding bones with diagnostic damage from the other species, a rare occurrence. One remarkable discovery from South Africa provides circumstantial evidence for such interactions. A fragmentary hominin skull shows unusual damage patterns that some researchers interpret as bear tooth marks, though this identification remains controversial. If correct, it suggests bears occasionally predated on hominins, viewing them as potential prey rather than competitors to avoid. Adult hominins were probably too dangerous to attack regularly. Groups could mob and drive off even large predators, but solitary individuals or youngsters might have been vulnerable. Modern bears occasionally prey on primates when opportunities arise, suggesting ancestral bears could have done the same with early hominins. More commonly, competition centered on carcasses. Both hominins and bears scavenge kills left by large predators, racing to find and exploit carcasses before they were completely consumed or decomposed. Evidence from cut-marked bones and stone tools shows early hominins actively accessed carcasses using tools to remove meat and break bones for marrow, resources bears also targeted. At sites where both hominin tools and bear fossils appear together, researchers can sometimes sequence events. First, a large carnivore kills prey. Then hominins arrive with stone tools to defleshing. Finally, bears appear and crush remaining bones for marrow. This temporal sequence suggests bears might have followed hominins, learning that tool-using apes led to feeding opportunities, or that both species independently located carcasses through scent and converged on them from different directions. The emergence of Homo erectus around 2 million years ago transformed hominin-bear dynamics fundamentally. These humans were taller, had larger brains, used more sophisticated tools, controlled fire, and hunted cooperatively. They were no longer just another primate competing with bears. They were apex predators reshaping ecosystems. Fire alone dramatically altered the relationship. Bears are generally fire-averse, avoiding burned areas and fleeing from flames. Humans who could create and control fire could clear bears from territory, defend kills from scavenging bears, and make caves and rock shelters unusable by denning bears. Archaeological sites from this period increasingly show evidence of regular fire use, coinciding with declines in local carnivore diversity. Tool technology gave humans additional advantages. Spears, even simple ones, allowed coordinated groups to kill dangerous animals from a distance. Evidence of early hominin hunting shows they targeted large herbivores, but also suggests opportunistic killing of carnivores when advantageous. Bear hides, meat and fat would have been valuable resources, potentially motivating humans to hunt bears directly rather than simply competing with them. Unlike ambush predators that relied on surprise and individual prowess, humans could coordinate attacks, wear down prey through persistence, and use terrain to their advantage in ways bears couldn't counter. Perhaps most significant was human capacity for learning, culture, and information transmission. Individual bears learned through trial and error over lifetimes. Human groups accumulated knowledge across generations, developing increasingly sophisticated strategies for exploiting resources and eliminating competitors. If one group discovered an effective technique for driving bears from territory or raiding their dens, that knowledge spread through cultural transmission, amplifying its impact far beyond the original innovators. Bears lacked any mechanism to counter this cumulative cultural evolution. Fossil evidence for human-caused bear declines remains indirect but suggestive. In regions where human populations expanded, bear fossil frequencies declined in subsequent time periods. The correlation is imperfect. Climate change and other factors also shaped carnivore abundances. But the pattern is consistent across multiple regions and time periods. Where humans arrived, large carnivores declined and bears were among the species affected. The mechanism might have been direct hunting, might have been displacement from key habitats, might have been disruption of prey populations through human hunting, or most likely was some combination of all these factors working synergistically. One possible scenario suggests human expansion into montane refuges delivered the final blow to declining bear populations. As African climates dried during the late Pleistocene, both humans and bears concentrated in remaining productive areas, mountains, rift valleys, coastlines. These refuges couldn't support both increasing human populations and their remaining bear competitors. Humans, with superior technology and social organization, monopolized the best territories and resources. Bears were pushed into marginal habitats where populations fragmented and declined toward extinction. The last bears might have died not through direct hunting, but through displacement into environments where they couldn't meet their basic survival needs. Modern ecological theory suggests another possibility: disease. Humans carried pathogens that could jump to other carnivores, and domesticated dogs, present in Africa by the late Pleistocene, hosted additional diseases transmissible to wild carnivores. Bears, with relatively small and fragmented populations, would have been vulnerable to emerging diseases in ways that large, wide-ranging carnivore populations were not. A single introduced pathogen could have swept through bear populations, pushing already stressed groups over the edge into extinction. While direct evidence for such disease-driven declines is absent from the fossil record, pathogens rarely fossilize. The scenario remains plausible and might explain the apparent rapidity of final bear extinctions. The relationship between early humans and African bears illustrates a broader pattern in evolutionary history. When new, ecologically flexible species evolve or arrive, they often drive established specialists toward extinction through mechanisms more subtle than direct competition. Humans didn't necessarily outcompete bears in any single niche. They weren't better desert survivors, better tree climbers, better bone crushers. But humans could modify environments in ways that made all those specializations less viable. They could learn and adapt faster than natural selection could reshape bear populations. They could cooperate across groups in ways solitary bears never could. The extinction of African bears may have been less about losing specific competitive interactions, and more about being unable to cope with a world increasingly shaped by creatures that changed the rules faster than evolution could respond.